In this video, we will cover the different stages of structure a protein takes. Although all proteins are unique in shape and function, they all follow similar processes of development into their final forms. Let's first discuss the building blocks of proteins. Proteins are made of amino acids. An amino acid is made of a central carbon atom linked to a hydrogen atom, carboxyl group, amino group, and an R group. There are 20 different amino acids commonly seen in protein. The R group, which is often referred to as the side chain, is where the amino acids differ. The side chain of an amino acid can be further organized into 4 categories. They can be polar, nonpolar, charged and acidic, or charged and basic. If the side chain is charged, it will be capable of forming ionic bonds. Polar side chains have partial charges and are therefore likely to form hydrogen bonds. Nonpolar side chains have no charge and are less likely to interact. The different types of amino acids used will later determine the final structure and function of the protein based on the types of interactions they can make. The primary structure of a protein is its unique sequence of amino acids. The amino acids are connected by peptide bonds. Peptide bonds are covalent bonds between the carboxyl group of one amino acid and the amino group of another. H2O is removed in the process, making it a carbon-nitrogen bond. These bonds are slightly stronger than most covalent bonds because the nitrogen partially shares its electrons with the nearby oxygen, giving it characteristics of a double bond and decreasing its flexibility. The long chain of amino acids is called a polypeptide. Polypeptides have 3 important features. The side chains extend from the peptide backbone, allowing them to interact with each other. The chains are also flexible everywhere but at the peptide bonds. Finally, the polypeptide chains have directionality. They start with an amino group called the N-terminus and end with a carboxyl group called the C-terminus. Once the polypeptide chain is fully formed, the primary structure is complete. Proteins, however, don't stay as straight chains. The chains are flexible and bend in space. This allows interactions between amino acids in the form of hydrogen bonding. Hydrogen bonds form when the oxygen of one amino acid comes close to a hydrogen of another amino acid. This creates a protein's secondary structure. There are 2 types of secondary structures. The first is an alpha helix, where the peptide backbone coils. The second is called a beta-plated sheet, where segments of the chain bend 180 degrees and then fold on the same plane. The type of secondary structure that forms depends on the sequence of amino acids in the primary structure. Proteins don't stop forming there. They continue to fold and bend in space, giving opportunity for even more interactions to occur. This time, from amino acids a great distance from each other on the chain. When a polypeptide chain is stabilized by these new interactions, it reaches its tertiary structure. There are 5 interactions that occur to make a protein's tertiary structure. Hydrogen bonds form between polar R groups, as well as on the backbone. Van der Waals attractions can take place when nonpolar side chains come together. The constant motion of electrons creates a weak electrical attraction between them. This interaction is made possible from hydrophobic interactions that push the nonpolar side chains together. There can also be ionic bonding between the charged R groups. Finally, there can be disulfide covalent bonds between 2 sulfurs from certain side chains. All of these create a much more stable structure for protein. The final stage of development for protein is the quaternary structure. Unlike the past 3 structures, this final stage includes more than one polypeptide chain. This stage is made possible when like polypeptide chains converge using the same interactions discussed for tertiary structure. The shape of the quaternary structure of a protein will determine its function, but said shape itself can be changed by a single swap of an amino acid during the primary structure. This accounts for the massive diversity seen in protein.